Assalamualaikum and hi everyone. It's me again, Madam Naziha binti Zainal from Biology Unit KMM. For this session, we are going to continue the last chapter, Chapter 9, Growth. For this chapter, there are two subtopics, 9.1 Measurement of Growth and 9.2 Growth Curve. But we will only cover 9.1 Measurement of Growth for this session. Our objectives are, first, you need to define growth. Second, state the three phases of growth, which are cell division, cell enlargement, and cell differentiation. Third, you need to state the parameters of growth, such as wet mass, dry mass, and length. What is growth? Growth is any permanent and irreversible increase in a quantitative parameters over a specific time. Quantitative means measurable, something that can be measured. Quantitative parameters are such as volume, width, length, height, cell number, dry mass, surface area, and biomass. And for example, we can measure length over specific time for measuring growth. There are three phases of growth. First is cell division. Second, cell enlargement. And third is cell differentiation. Cell division is the basic process in growth. Cells divide to form new cells by mitosis to increase the number of cells. During cell enlargement, it will increase in size and volume of cell because increase of cytoplasmic volume and size of nucleus. This is because the cell increase in the number of organelles. Each cell grow to the limit and the cell divide before it reach the limit. What is limit? Limit is the ability of the cell nucleus to support the increase in cytoplasmic volume. This is example cell enlargement in plant cell. Cells grow by adding organic material to their cytoplasm or addition of water into the large central vacuole. This accounts for 90% of plant cell expansion. As you can see in this picture, plant cell will expand when water is added. And this is example cell enlargement in animal cell. Cells grow by synthesizing a protein-rich cytoplasm, which is a metabolically active process. Cell differentiation is the changes in the cell form, either shape or structure for specialized function. Thus, a cell divides repeatedly, resulting in a group of cells that are different from each other in its structure and chemical composition. Example cell differentiation in plant cell, where meristematic cell differentiate into xylem for transportation of water and minerals, and meristem cell differentiate into phloem for transportation of product of photosynthesis and also hormone. While cell differentiation in animal cell, where red blood cell lost their nucleus to provide more space for hemoglobin, thus enhancing the ability for transporting oxygen and carbon dioxide. Okay, next, you need to know three types parameters of growth. First, wet mass. Second, dry mass. And third, length. All these are quantitative parameters, meaning that parameters that can be measured. For example, you can measure your height, measure your length of hand, and measure the length and width of an animal. And for biomass, we can measure the total mass of all living material in a specific habitat. For wet or fresh mass method, this is the most appropriate method to measure growth of a multicellular organism. The mass is obtained without the removal of internal water, meaning that in the normal condition. Advantages for using this method. First is easier because no complex preparation required. Second, we do not need to kill the organism and the growth can be measured continuously. And last one, same organism can be used repeatedly. But there are some disadvantages for using this method. First, it is inaccurate and inconsistent due to the fluctuation of water content in the organism. Water content will increase or decrease regularly due to the water uptake or water removal by the organism, and less suitable to be applied for living tree. But the problem can be solved by using dry mass method. Second method is dry mass method. The mass is obtained after all water is removed through drying method. An advantage for this method is more accurate. First, we need to kill the organism. Second, heat in the oven. Third, cool in the desiccators. And fourth, weigh it. We need to repeat step number 2 to step number 4 until we get constant mass. But there are some disadvantages for using this method. First, the organism is killed, so we cannot measure further growth. Second, time-consuming process because we need to repeat step number 2 until step number 4. Third, in repeated measurement, we will require larger sample size of organism with similar size and age as representative reflection of growth. And last, limitation occurs because it's not applicable to animal. Question time. Try to answer this question. Number 1, define growth. Number 2, state 3 phases of growth. And number 3, state 3 growth parameters.